Hello, everybody. Welcome back. We are continuing our journey with our harvesting energy chapters. We are now on to chapter 8, which is going to discuss glycolysis and cellular respiration. Just to recap, we just got done with chapter 7, which was dealing with photosynthesis. So we are going to be seeing some similarities in some of these processes that are taking place inside of the mitochondria, similar to some of the processes that took place in the chloroplast. So there was a little bit of a discussion about sugar and water and oxygen and carbon dioxide in that photosynthesis lecture. We're going to see that relationship again here in chapter 8. So remember, they're kind of like the reverse of each other. As water and carbon dioxide is used to make oxygen and sugar, we're going to see in cellular respiration that sugar and oxygen is broken down into carbon dioxide and water with the release of energy. So that's kind of our main goal, is to try to explain those things. So let's go ahead and get started. Okay, so we're starting our question or our lecture out with this question of how do cells obtain energy? So there's a couple different ways, right? So photosynthetic cells obtain energy by photosynthesis, right? So they use the power of the sun's energy to convert carbon dioxide into sugar, and that sugar is that stored potential chemical energy that they can then send on to their mitochondria. Now, if you are a heterotroph, like a human or an animal or a fungus, you have to, or a protist, you have to take in nutrients or your sugar from consuming other things. You do not have the ability to make your own sugar. So that is how cells obtain energy. And ultimately, with the exception of those hydrothermal vents, got to do a plug for those because of my oceanography class. So the ultimate source for almost all life on Earth is the sun, because we are either going to be getting that sugar via photosynthesis or eating that sugar from organisms that do photosynthesis. So just a reminder, we see the equation here on the slide of photosynthesis. We have carbon dioxide and water, gives you sugar and oxygen, with light and chlorophyll kind of hovering over those reaction arrows. So I just have a picture of a plant and some photosynthetic algae, since it's really the unicellular organisms on this planet that produce way more oxygen than land plants. So ocean ecosystems are super valuable for the production of oxygen that we need in our atmosphere. All right, so with glucose, we know that glucose is a primary product of photosynthesis. And if you remember from our biological molecules chapter, carbohydrates, glucose is a monosaccharide, part of the carbohydrate group. Their primary job is to be an energy storing molecule. So the energy from the sun is stored as potential energy in those chemical bonds. And then cells can then break that down to release it into more usable forms of energy like ATP. This breakdown is fairly simple, and we'll be seeing a little bit later that other nutrients like the fats and proteins can also be metabolized, very similar to glucose. But what I've done here is I've showed some different representations of how you might be seeing the molecule of glucose in this lecture and your textbook and animations, just so you can kind of see that it's all the same thing. So glucose, remember, is C6H12O6. It can be in a couple different forms. We can have it in the linear form, which is this one right here. Sorry, that's the ring form. So here's our carbons. We have 1, 2, 3, 4, 5, 6 carbons. If we're to count the oxygens, we have 1, 2, 3, 4, 5, 6 oxygens, and I'll let you count all 12 hydrogens. So everything's accounted for, and in this particular case, it kind of folds into this ring. It's called a hexagon or a hexose sugar because it makes this 6-sided ring. Now in this particular, this is the linear form now. You might be saying, well, Corey, I only see 1 carbon. Where's the other 5 carbons? Well, in organic chemistry, when you are drawing the molecules in organic chemistry, a lot of times you can take shortcuts. So sometimes I like to say, joking around, chemists are lazy. They like to take shortcuts. So wherever there's a corner or a cross of 2 lines, this is representing where a carbon atom would be. So instead of drawing them, we're just assuming that that's where the carbon is going to be. And remember, carbon had 4 valence electrons, so it can bind to 4 things. So we should be able to count that every carbon has 4 bonds to it. So we have 1, 2, 3, 4, 1, 2, 3, 4, and so forth and so on. So we should be able to account for all of that. Oxygen has 2, hydrogen has 1. So that's just kind of reinforcing what we learned back in chapter 3. A more simplistic way that you might be seeing glucose is kind of in this just a carbon skeleton. The oxygens aren't shown, the hydrogens aren't shown. And a lot of times when you see it in this way, you're just trying to focus on keeping track of those carbons. We'll be seeing this in the glycolysis section in the Krebs cycle, since that's where we're going to be doing most of our glucose metabolism. So whenever you see carbons, usually in a black ball with a C labeled on it, it's just showing you that those are saying there's carbons. There might be other things hooked to them, but that's not really what's important at that particular picture or diagram, just that you're keeping track of those carbons. So you'll be seeing glucose portrayed in multiple different ways as you're going through these topics. Okay, so glucose has a lot of cellular energy stored in those covalent bonds, right? The 6 carbons, 6 oxygens, 12 hydrogens. But it's really hard, like we talked about before. We can't just eat a banana and have the banana nutrients go and power our muscles. All of our cells are kind of programmed to use one common currency in the cell, which is ATP. So sometimes it's called the energy currency of the cell, cash currency of the cell. Just like in the analogy here, you can go into a store. The store might be able to take checks or credit cards or cash. Cash is almost taken everywhere. It's readily available and it's easy for the transaction. So you can think of ATP as kind of like the cell's energy cache, if you will. Okay, so just like we had an overview or a summary equation of photosynthesis, we have a similar type of equation for cellular respiration. And if you take a look at it, it looks like just the reverse of photosynthesis. So let's take a look at what we have. We have our glucose, C6H12O6. We have some oxygen, okay? So here's our reaction arrow. So this is actually a whole lot of steps that we'll be diving into in a little bit. And then here's our product of carbon dioxide, water, and then the energy. So if you remember, photosynthesis was the reverse of this. We took water and carbon dioxide through a whole bunch of reactions. We produced oxygen and sugar, and the energy was an input with the sunlight, right? So that was an endergonic process to produce a more complex glucose molecule from less complex carbon dioxide molecules. Cellular respiration is an exergonic reaction. We are taking a large complex glucose molecule and breaking it down into smaller, less complex carbon dioxide molecules. So this is a exergonic releasing of an energy process, and there's the ATP to account for that. And so now we have this usable form, this cash currency of ATP in our cell, which can then go and do a whole bunch of things. And so for this last bullet point, so just like we took photosynthesis and we had to break it down into all of the different things that happen in the light reaction and all the different things that happen in the Calvin-Benson, so the Calvin cycle, it's the same kind of idea with glucose breakdown, this cellular respiration. It's not just one step, it's a lot of steps. We have a couple different phases of glycolysis. We have a couple different stages of cellular respiration. And so we'll just kind of work through each one of these individually. Okay, so when we take a look at glucose breakdown, sometimes it's called metabolism or catabolism. So when you're talking about metabolic processes, when you do catabolic pathways, you're breaking things down. So we're going to do glucose catabolism or just metabolism, which is the chemical reactions. It is broken up into kind of 2 phases or 2 stages. We have the glycolysis part. And if you take a look at the root words, right? So glycolysis, we can split it right down the middle. Glyco means sugar, and lysis, do you guys remember what lysis means? We did in hydrolysis reactions. It means to split. So glycolysis actually means sugar splitting. And as we go through, you'll see, oh yeah, it does. We actually take glucose and we split it into 2 smaller molecules. So this happens within a cell in the cytoplasm. So this does not need a special organelle to happen. So glycolysis, all of the enzymes that are used in this reaction are found in the cytoplasm of eukaryotic cells, plant cells, animal cells, fungal cells, protocells, It is also common to all bacteria, archaea, and bacteria. So this is a very common process found in all living cells. We share this ability to harness the energy from the glucose to turn it into ATP. This is an anaerobic process, which means it does not require oxygen. So the splitting of sugar just needs glucose and a whole bunch of enzymes and a little bit of ATP to start the process, right? We've got that activation energy we have to overcome. All right, so then the second half, if you will, of glucose metabolism is what we call cellular respiration. So glycolysis starts it, and then in the presence of oxygen, we can finish this breakdown of glucose to try to get as much of that energy, that potential energy stored in those bonds out as possible. This process takes place in the mitochondria of eukaryotic cells. It does require oxygen, but prokaryotic cells do cellular respiration as well. They just don't have a mitochondria, so all of their processes have to happen on their plasma membrane and within their cytoplasm. All right, so here is a kind of overview picture of what we're going to be discussing in this chapter. So we are always going to start with glucose, right? So that's our starting point. In the process of glycolysis, we're just going to see a very basic version of it. But if you're to go into a lot more detail, there's actually 10 individual steps of glycolysis. We're just going to kind of split it down into 2. So then you get the product of glucose, which are a couple molecules called pyruvate or pyruvic acid. They're basically the same thing. So you get some carbon-containing molecules, that's the pyruvate at the end, and you get a little bit of ATP, just a couple ATPs in the process of glycolysis. If there's no oxygen available, that pyruvate can be processed in fermentation. That's gonna wrap up this chapter. We're not gonna talk about that until the end. And that could either produce a lactate or lactic acid or ethanol, which is a type of alcohol. So we'll see that a little bit later towards the end of this chapter, where cells can utilize pyruvate in the absence of oxygen to still carry out their metabolic processes. If oxygen is available in our aerobic oxygen-breathing organisms, then that pyruvate goes into the mitochondria, excuse me, to finish this process of complete breakdown into the reactions of cellular respiration. And this is where we need our reactant of oxygen. This is where we get our products of carbon dioxide and water, and our also product of ATP. And if you'll notice, there's a lot more ATP produced in cellular respiration than in our beginning processes of glycolysis. We get a little bit of ATP to start us off, but we really want oxygen to give us the most bang for our buck, if you will, when we're talking about extracting that stored energy out of glucose. All right, so let's dive into this first sugar splitting, right? So glycolysis, glycolysis. So there's 2 phases. So the first one is glucose activation, or you can call it the energy investment phase. This is kind of getting everything started. So here's those 6 carbons of glucose represented. We're ignoring the oxygens and hydrogens at the moment. So we have to put in a little bit of energy to get this process started. So we saw that with our activation energies, and enzymes kind of reduce that activation energy. So for even exergonic reactions that are kind of spontaneously occurring, you will release energy. We have to get a little bit of push to get things going. So we burn a couple ATPs to get the things going. So it's kind of like when you're making money. You have to spend money to make money, that investment phase. So we break apart a couple ATPs, we hook those phosphate groups onto the glucose, we rearrange the carbon molecules a little bit and convert it from the glucose that we started with to something that's called 1,6, fructose bisphosphate. So what that means is we have a fructose molecule with 2 phosphate groups hooked onto it. One's hooked onto the number 1 carbon, one will be hooked on to the number 6 carbon. Okay, the second half of glycolysis is a little bit more detailed. So I'm going to have 2 slides to go through all the steps that are happening here. Lots of arrows are going all over the place. So in the second half, it's called the energy harvesting phase. So we spent a little bit of our ATP, but we're going to get a bigger payoff at the end of this process. So that that fructose bisphosphate, it was a 6 carbon molecule, is split into 2 3 carbon molecules. So we haven't lost any carbon, we're just splitting it, right? There's our water, there's our glycolysis. We've split our glucose. So now we are having 2 3 carbon molecules called glyceraldehyde 3-phosphate. Hey, that might ring a bell. We just got done talking about that in photosynthesis. So that was part of the Calvin cycle, where as the carbon dioxide was coming in, it spun around and it kicked out 1 glyceraldehyde 3-phosphate for every turn, 2 G3Ps hooked together to give you a glucose. So we're almost doing the reverse of what we saw in the Calvin cycle of photosynthesis. There was overlap, right? We knew that was going to happen. So that 1,6 bisphosphate, fructose bisphosphate, is split into 2 G3Ps. And so we still have our phosphates. We haven't done anything with them. So here we have one of the phosphates from 1 ATP. Here's the other phosphates, and we still have 6 carbons. All right. So this step here, lots of things are happening. So one, we have the blue arrow is going to be representing what's going on with the carbon. We're rearranging. The enzymes are in there rearranging these carbons. So they just change their identity from what we call glyceraldehyde 3-phosphate to pyruvate or pyruvic acid. All right, so that's the blue arrow is showing you the change of the carbon molecule. The arrow on the top is showing you how we now can take our ADPs, right? That's the diphosphate. We are going to start cramming that 3rd phosphate group on to our ATPs. So we are going to be making 4 ATPs in this energy harvesting phase. Now, so we're making 4 total. We spent 2, so that gives us a net gain of 2 ATPs. So in glycolysis, you actually generate 4 ATPs, but we have to account for the 2 that we spent at the beginning. So we usually say glycolysis has a net gain of 2 ATPs. All right, so for every glucose molecule that goes into glycolysis, we get 2 pyruvic acids, we get 2 net ATPs, and then this last arrow here, we get, we're stripping some of those electrons away, right? We know these electron carriers temporarily hold on to these high energy electrons. So we have a couple NAD pluses with some hydrogen that's coming with those electrons to create 2 NADHs, all right? So that's the process. So I have it written here in the text, and that's there in the picture, and I think I continue this process on the next slide. Yeah. So there's our G3Ps, glyceraldehyde 3-phosphates. We strip some of those electrons and put them on the NADHs to make NAD pluses to make NADHs. And so now we get our end result of glycolysis, 2 net ATPs, 2 pyruvic acids, and 2 NADHs. So let's see it all put together. So here's the entire process. Now, like I said earlier, in reality, this is broken up into 10 individual steps. Each step is catalyzed by a different enzyme, and you need to have all of those enzymes for these processes to work. But for this class, we just kind of focus on these 2 parts. These 2 major parts is we have our energy investment phase where we phosphorylate, which means adding the phosphate groups to our glucose. So we kind of get things going, and then we harvest that energy by stripping those phosphates to make 4 total ATPs, 2 net ATPs. We strip some electrons and tag them on to the NAD to make our NADH. And now we have still 6 carbons. We haven't lost any carbons at this point, but we've harvested a little bit of energy out of them, right? We've got a couple ATPs, we've got a couple NADHs, right? So that's our first part of all of these processes is glycolysis, right? But that's not a lot of energy. 4 ATPs, you'd think glucose would have a lot more than that. And it does. We got to move on to the mitochondria because remember, this is happening in the cytoplasm. All right, so here's our summary. I just found a little picture, slightly different from your text. It's always good to see things that are in a little bit of a different way. So here we are seeing our 6 carbons in glucose. We have our 6 carbons, but now in the form of 2 pyruvic acids. We have hooked our electrons onto our 2 electron carriers, and we have our 2 net ATPs. Okay? So you should be comfortable with these kind of what goes in, what comes out, and kind of the purpose of this process of glycolysis is to start this harvesting of energy of those electrons that are storing that energy in those covalent bonds. Okay, so now we're moving on. So we got to do something with that pyruvic acid. We've made a couple ATPs, we've got a couple NADHs, but that's not enough to do most cellular processes. So for aerobic organisms, organisms that use oxygen, those pyruvates still have quite a bit of energy stored in them, those 3 carbon molecules. We can't just be done there. We can take those and further break them down. So this is what takes place in the mitochondria. So those pyruvates that are made in the cytoplasm will go into the mitochondria and then will be further acted upon in there. So we got to remember a little bit about what the mitochondria looks like. So I have on the next page just a picture. Remember, our mitochondria was an organelle, kind of looks like a jelly bean. It had 2 layers. It had the outer membrane, which separates the organelle from the cytoplasm and everything else. And then inside we have the inner membrane, which was wrinkly. It had all of those wrinkles in it, if you remember, that helped to increase surface area. And now you'll see why as we're going to dive into what's actually happening on this membrane. So we've got 2 membranes. We've got an outer membrane, we've got an inner membrane. And so now we have some compartmentalized spaces. So between the outer membrane and the inner membrane, we have what's called the intermembrane space. So this is going to be really important for setting up a concentration gradient, which is another crossover with photosynthesis. We're going to be seeing a hydrogen ion gradient getting formed in the last stage of cellular respiration. And then we have the inside space inside that inner membrane, and this is called the matrix. So this is where what we call the acetyl-CoA synthesis and Krebs cycle are going to be taking place inside the matrix. The intermembrane space is where we're going to have our hydrogen ion concentration, and then the actual membrane itself, this is where ATP is going to be produced with proteins embedded on that inner membrane, very much like the electron transport chain embedded on the thylakoid membranes of the chloroplast. So again, lots of similarities between chloroplast and mitochondria structurally and functionally. And then up here is a picture of electron micrograph of an actual mitochondria. So you can see the outer membrane, you can see the inner membrane with all the folds, the space here is the matrix. Here's a crista, which is the wrinkles, okay? And the little inner membrane space. If you look really closely, you can see that gap there between that inner membrane. Okay, so just like glycolysis had 2 phases, we had the energy input and then the energy harvesting phase. Cellular respiration also takes place in kind of 2 stages. The first one is got 2 parts to it. So the acetyl-CoA production and Krebs. This takes place in the very center part of the mitochondria. That's what we call the matrix. And then embedded in that inner membrane, this is our electron transport chain. So we're going to attack cellular respiration, taking a look at those 2 components. So we have what's happening in the matrix with this short little acetyl-CoA step in a Krebs cycle, and then what happens on the inner membrane with the electron transport chain. Okay, so the first part of what's happening in the matrix is the acetyl-CoA. So the pyruvate that was produced in the cytoplasm at the end of glycolysis moves into the mitochondria through the outer membrane, through the inner membrane, into the matrix, right? So here's our pyruvate. Remember, it was a 3 carbon molecule, and we get 2 of those for every glucose that goes into glycolysis, okay? So here we have another really busy arrow. A lot of stuff is happening. So we'll kind of go piece by piece. So this main gray arrow in the middle, right, takes a look at what's happening to the carbons. So if we take a look, we have a 3 carbon pyruvate is changed into a 2 carbon acetyl molecule. But take a look, that gray arrow splits kind of halfway through because, take a look, let's keep track of our carbons. If we start off with 3 in pyruvate, we have now 2 in this acetyl molecule. We lost a carbon somewhere. And that's what this arrow here is showing you, is we actually lose a carbon dioxide. So we go from a 3 carbon pyruvate to a 2 carbon acetyl molecule. And so one of those carbons is lost as carbon dioxide. That's actually some of the CO2 that you exhale. So when you talk about respiratory gases, oxygen goes in and carbon dioxide comes out. This is where the carbon dioxide is coming from. Isn't that cool? You're learning about your body inside your cells. That's why I love science. Okay, so let's get back to it. So the gray arrows are showing you what's happening to the carbons. We start off with 3, we end up with 2, we've lost 1 to our waste products. Now this arrow here, the coenzyme A, coenzymes are kind of like enzyme helpers. So they allow for the enzyme to work more efficiently and better. It kind of helps with that substrate active site connection, right? That complex it's formed. So as we exchange, we lose one of the carbons, we are going to tag that coenzyme onto that 2-carbon molecule. So the acetyl group is now going to become acetyl-CoA. Okay, so this is what we call the acetyl-CoA production, or acetyl-CoA step is what I call it in my biology classes. So then the third thing, so there's 3 things that are happening in this step. We have the conversion of pyruvate to acetyl-CoA. We have the losing of one of the carbon dioxides. And then our last step is, take a look. We have another one of these electron transfer reactions. So we've got an NAD+. We are going to strip some electrons away from that pyruvate and turn it into NAD+. Sorry about that. The cat just stepped on the keyboard. Say hi, kitty. I'm being a pest. Okay, sorry. Let's get back on track. Okay, so the NAD+ is going to be stripping the electrons away from the pyruvate as it's doing all of these other things. So as a pyruvate is turning into acetyl-CoA, it's losing the carbon dioxide, it's hooking onto that coenzyme A. We are stripping some of those electrons, hooking them to NAD+ to create our NADH. Okay, so at the end of this kind of little mini step, lots of stuff is happening. We produce 1 carbon dioxide that is lost as waste. We produce 1 acetyl-CoA molecule, which will head into the next step, and we have 1 NADH. Right? So we are slowly building up those electron carriers. We made a couple with glycolysis. We're going to make a couple with the acetyl-CoA production because there's 2 pyruvates per glucose, right? So we have a couple NADHs, a couple carbon dioxides, a couple acetyl-CoAs. Right? So that's the first part of this stage 1 of cellular respiration. Okay, so then that moves us into what's called the Krebs cycle. This is also known as the TCA cycle or the citric acid cycle. So if you see animations or tutorials elsewhere, they might call it something different. I like to call it Krebs because it's easier to say than TCA, which stands for tricarboxylic acid. That's a mouthful, or citric acid cycle. So we're just going to call it Krebs. So we're still in the matrix of the mitochondria. We have our acetyl-CoA now is produced, and that's going to trigger the first part in this. Again, it's a cycle, just like the Calvin cycle to produce glucose. This is a cycle which is helping us further break down glucose. So again, it's a long series of reactions. We're just going to kind of cover the basics here. But look at our major products. So as we are going around the circle, we quickly kick off that CoA. It's done its job. It's shuttled that acetyl group, that 2-carbon group, into this series of chemical reactions. We are going to do another production of NADH, another production of a kind of a cousin of NADH called FADH2. We are going to get rid of some more carbon dioxide. We exhale that as gas, and we make a little bit of ATP, 1 molecule of ATP per spin. Okay, so the things that I've circled are the products of the Krebs cycle. So we have 3 NADHs, 1 FADH2, 2 carbon dioxides, and 1 ATP. Okay, so that is the product of 1 spin, and we get 2 spins per glucose molecule, right? Because each glucose produced 2 pyruvates. The 2 pyruvates will produce 2 acetyl-CoAs. Each acetyl-CoA will spin the Krebs each time, so 2 total. So by the time we get done with glycolysis, the acetyl-CoA step, and Krebs, we have got a whole pile of NADHs. We've got a couple FADH2s and a little bit of ATP. But really what we are wanting is all of those high energy electrons that are stored in the NADH and the FADH2, because they're going to do work for us. They are potential energy. We're going to go and send them to the electron transport chain and make a whole lot of ATP. And here's some more CO2 as our exhaled waste product from just living, from our cells being cells. All right, so here's all of those put together, what we call the stage 1 reactions that are happening in the matrix of the mitochondria. So we have our pyruvate, which was produced in the cytoplasm, diffuses into the mitochondria, It's converted to acetyl-CoA. We get rid of some CO2. We collect some electrons in our NADH. The acetyl-CoA moves into these series of Krebs cycle reactions. We produce more NADH, FADH2. We give off more carbon dioxide as waste product, and we have our ATP. Okay, so if you've been keeping track of your carbons, now it's kind of tricky because you got to double everything per pyruvate. We should be accounting that we have exhaled 6 carbon dioxides because we started with 6 carbons in glucose, right? If we look at the reaction or the equation, C6H12O6 goes to 6 carbon dioxides. So let's see if we can count them per pyruvate, and then we'll double it, right? So we have 1 carbon exhaled in the acetyl-CoA step, and we have 2 carbons exhaled in the Krebs cycle. So with 1 pyruvate, we have given off 3 carbons in the form of carbon dioxide. So there's 3 in 1 pyruvate, and we have 3, if it to do all of this again, 6. We've just completely metabolized or broken down glucose into that final, very minimally energy-rich molecule of carbon dioxide. So we are done with glucose. We have stripped as much of its energy away as possible. Where did that energy go? Well, we haven't made ATP. We've done a little bit of ATP. We've made 2 ATP in glycolysis. We've made 2 ATP with 2 spins of Krebs, 4 ATP. That's a little bit low from 1 molecule of glucose. Where is some more energy outside of ATP? Well, if you remember, we have 1 NADH was made here. We have 3 NADHs here. We had 2 from glycolysis. Oh, that's not looking good, right? And we have an FADH2. So if we double everything that's on the bottom, so that's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 NADHs and 2 FADH2s by the time we're done with all of glycolysis and these stage 1 reactions, which means we have a lot of energy, high energy electrons in these electron carriers, whether it's that mailman with the satchel that we saw earlier or an electron bucket, right? So these NADHs, FADH2s, think of them as these temporary electron carriers. They're just holding on to these electrons, taking them from the matrix and the cytoplasm, right? The ones that are producing glycolysis. And now we are going to take them to the electron transport chain because that's where we're going to get most of our work done. Okay, so lots of words here, and then we'll kind of work through the picture. So at this point, most of the energy in glucose has been stored in these NADHs and FADH2s. We've made 4 ATPs from glycolysis and Krebs. And so now we are going to start utilizing those electron carriers, the NADH and the FADH2. They're going to move from the matrix towards the inner membrane, the little wrinkly one, that inner membrane in the mitochondria. And eventually what's going to happen, we're going to dump those electrons, allow them to move through the chain. And at the very end of this electron transport chain, those electrons are going to bind with oxygen, what we breathe in, with some hydrogen ions, and we're going to make water. So that was one of our products, right? So glucose plus oxygen gives you carbon dioxide and water. We've accounted for the glucose and the carbon dioxide. We've just accounted for the oxygen needing to make the water, which is the other product, and then all of those ATPs. So we're going to bring some of our prior knowledge back. So we are going to take a look at the phospholipid bilayer and how it's selectively permeable. We're going to see some very specific transport proteins like what we saw in the thylakoid membrane in photosynthesis. We're going to see concentration gradients, active transport, passive transport, and a reaction coupling. So the idea of an exergonic reaction to drive endergonic reactions. So right, culmination, all of that prior knowledge from chapters 5 and 6. So membrane structure and function and energetics and enzymes, it's all coming home to hopefully make sense in these cellular processes. All right, so let's take a look. This is a very busy picture, so I'll try my best not to make it even more messy looking. So we'll take it step by step because I know it can be a little bit overwhelming. You have your picture of this process on page 119 of the new edition of the textbook. So again, location, where are we? Let's kind of orient ourselves. So here's our picture of the mitochondria. we are actually looking at this inner membrane. So if we're to zoom in, here we have our inner membrane space. Okay, so that's a little space between the outer membrane and the inner membrane. And then here's the matrix. So this is where your Krebs cycle is taking place. This is where your NADHs are produced, your FADH2s. So that's why we're going to be seeing these over here on this side of the membrane, because that's where they just got made there. All right, so that's where we're at. So you can kind of visually put yourself in that position in the cell. So here's our phospholipid bilayer, right? It's selectively permeable, so ions cannot move through it. We should be comfortable with that idea. And only certain things can move through it, or if we wanted to move an ion through, we have to have that specialized channel. And that's what we're going to be seeing here at the end with the ATP synthase. We saw an ATP synthase before in photosynthesis. So again, we're going to see some similarities between these two. All right, so the first thing that's going to happen is the NADH is going to come along, and it's going to actually reverse the reactions of how it was made. So those electrons that we got stripped away from the carbon molecules, we're just going to give those up. So the NADH releases the electrons and the hydrogen, because the electrons usually travel with the hydrogen. And so we are going to allow those electrons to kind of temporarily be held by these membrane proteins, the blue blobs, right? They're proteins. And they're enzymes at the same time. So they're kind of structural and they're functional, allowing these reactions to take place. So I'm going to follow the track of the electrons. The electrons are going to go here to this first carrier, and then they'll be carried through the membrane to the next carrier, and then through another carrier and another carrier and another carrier. And by the time we get done, we're going to hook those electrons with some hydrogen, right, and some oxygen and the electrons, and we're going to get our product of water. Okay, so that's the flow of the electrons. And it is very similar to what we saw in photosynthesis, where if you remember, when we dropped those electrons, or the sunlight energized those electrons, as they were going down their energy level, they were releasing energy. And then it's the low energy electrons that were picked up at the end of the electron transport chain. It's the same kind of idea. Now, FADH does the same thing, but it just releases its electrons at a different blue blob, right? So it just drops them off at the second stage. So here we have our NADHs, FADH2s. We've harnessed all of those high energy electrons from our glucose molecule. Now we're taking them to this inner membrane. We're dumping them into these blue blobs, into these transport proteins, or these membrane-bound proteins, and they're just going to be moving. They're just bopping from one to the next, from do, 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 do. And at the very end, that's where your oxygen is going to come in. So the oxygen that you breathe, take a breath. All of that oxygen that you're breathing into your body, carried by your blood out to all of your cells, this is its role. That's its only job, is to come in and to hook all of these things together, to take up those electrons that we pulled away from glucose, hook it up with some hydrogens and the oxygen we're breathing to produce water as one of our products. Okay, that's great. But that's not all, because remember, these were high energy electrons. And as they're moving through all of these transport proteins, they're giving off some of their energy because they're falling back down to a lower energy state. So because they have this ability to release energy, we're going to harness that energy and do work. So just like in the thylakoid membrane, we're going to be pumping protons. So all of these red arrows, let me get a different color here. So all of these red arrows, they are going to be representing all of the hydrogen ions that are going to be pumped into that inner membrane space, that little space between the inner membrane and the outer membrane. So take a look at our hydrogen ion concentration. Find where we have a lot and find where we have a little. Okay, so just look at the picture. We have more hydrogen ions in that inner membrane space and fewer hydrogen ions in the matrix. So if we are pumping hydrogen ions into the inner membrane space, we are going against their concentration gradient, which means it's active transport. And where do we get the energy to drive that active transport? Those electrons. Where do we get those electrons? From glucose. That's why we need to eat food. We have to eat food to get electrons to drive the electron transport chain, to pump protons, to make this concentration gradient. It's all coming together, right? All right, so now what are we going to do with this concentration gradient? Well, we're going to let it do work. This is a potential energy source. We have a whole bunch of hydrogens back behind the dam, right? They are impermeable. They cannot go through that phospholipid bilayer by themselves. They have to have a special channel. So it's this right here, this beautiful enzyme is called ATP synthase. It is permeable to hydrogen ions. So it's taking advantage of that concentration gradient. So as the hydrogen ions flow through, it's not shown in this picture. You might see it on some of the animations that you can watch. It actually spins. It is a physical motor. It's the same one that's in the chloroplast. It is a physical motor that as the hydrogen ions travel through, it spins, and it's that physically pushing that third phosphate group onto the ADP that allows you to generate ATP. So there's our reaction coupling. The reaction of the exergonic diffusion of hydrogen down its concentration gradient with the endergonic production of ATP, this hard third phosphate group to push on, and voila, we produce a lot of ATPs. So I think I have a picture of a counting of all these, yeah, when we kind of put it all together. So that's the second part, kind of stage 2 reactions of cellular respiration is the electron transport chain. All right, so again, we'll see some summary slides, but on this particular picture, kind of what goes in, what goes out, and why. Well, what goes in are the electrons from your electron carriers, the NADH, the FADH2. What comes out? Water and ATP. Why? Okay, or oxygen goes in too, so we can't forget about oxygen. So NADH, FADH2 drop off their electrons. Oxygen comes in from our breathing. Okay, what comes out is water as a byproduct of oxygen, hydrogen ions, and electrons being at the end of the electron transport chain. And then we have ATP being produced by the ATP synthase. Okay, so take a look at the summary of kind of all of these parts put together. So here we have in the cytoplasm, right? So that's the purple space. Here's our process, our first step. Oh, I'm still on my purple pen, cool. So here's our glucose, is broken down via the process of glycolysis into pyruvate, right? We haven't lost any carbons, but what we have done is we've produced some ATP and those high energy electron carriers, a couple NADHs, okay? In the presence of oxygen, the pyruvate goes into the matrix of the mitochondria to produce our acetyl-CoA, right? So what have we produced in this kind of intermediate step? We have a couple of those high energy electron carriers, and we have released a couple of our carbon dioxides, right? So part of our waste product. Okay, so we're still in the matrix. The acetyl-CoA goes into the Krebs cycle. We do a lot of reactions. We're stripping more and more of those electrons away from these carbon-based molecules. We produce a lot of NADHs, a couple FADH2s, and a dinky little 2 more ATPs, right? So again, we have 4 ATPs at this point. We have 1, 2, 3, 4, 10 NADHs, 2 FADH2s. That's where most of the energy is stored from the glucose molecules. We stripped it away in those forms of those high energy electrons, right? And then we give off some of our carbon dioxide, the rest of our carbon dioxide in the Krebs. All right, and then lastly, we come to the electron transport chain where all of our NADHs and FADH2s come down. They drop off their electrons to have the electrons pop down that electron transport chain, pumping the protons, making the gradient, and allowing those hydrogen ions to flow back down ATP synthase, physically spinning this little cellular motor to generate our ATPs. And look at how many ATPs we get from the electron transport chain. What? 32 ATPs per the breakdown products of 1 molecule of glucose. So if to add them all up, we get 32, 33, 34, 35, 36. So for 1 molecule of glucose, we can generate about 36 molecules of ATP. That's pretty impressive. But let me tell you something. Each one of your muscle cells burns about 5 to 10 million molecules of ATP per second. Right? Millions of molecules of ATP per second just for your muscles to do regular muscle-y stuff. So 1 glucose only gives us 36 molecules of ATP, which seems like a lot, but in context, That's why we have to have so much food coming in. We have to have that certain maintaining caloric intake to keep all of our cells readily available with enough ATP to do all of the things that we need to do. Pretty impressive. Okay, so we just got done talking about glucose and how it runs through cellular respiration. But we eat more than just glucose, right? We have other nutrients we saw back in our organic molecules chapter. So this center pathway, this is what we just covered. So carbohydrates, starches can be broken down into your simple sugars. They go through everything we just talked about: glycolysis, acetyl-CoA step, Krebs, electron transport chain, getting us ATP. There's a lot of people out there that are doing no carb diets, like the keto diet or the Atkins diet. So how do they get energy? Well, those diets are kind of focused on trying to get your body to burn fats. So that's why a lot of people lose weight. I have my own opinions about that. I'm not going to share that in this class. But you have fats from the food that you're eating, right? So if you remember, fats were made up of a glycerol, 3-carbon backbone with those big, long fatty acid chains. And so those can actually get broken into the intermediates. You guys remember G3P, glyceraldehyde 3-phosphate? It was a 3-carbon molecule. So glycerol can actually be converted into G3P. And so they can just shove that right into glycolysis and let it continue that process. Fatty acids can be broken into 2-carbon molecules, those little 2-carbon acetyl groups. So we can just shove those right into the acetyl-CoA, send them right into Krebs. So we can actually derive energy from the fats that we're eating. Excuse me. Proteins are also can be burnt for nutrients. We typically don't. We usually focus more on carbohydrates and fats. But if we need to, if our body is low on carbs and fats, we can use proteins as a food, as a nutrition energy source. It's not the best for you because there's more toxic kind of byproducts of protein metabolism. But remember, proteins are big, long chains of amino acids. So we can break those down into those individual amino acids. And then we have reactions inside mainly our liver and our skeletal muscles that can convert those amino acids into pyruvate, which can go into the system. They can be converted into acetyl-CoA, which can go into the system, or they can be converted into some of the intermediates that we didn't really see, but they can go into the Krebs cycle. So we, from all of our 3 organic macromolecules, proteins, carbs, and fats, all of them have the ability to go in and feed into glycolysis, the acetyl-CoA step, Krebs, to generate those electron carriers to drive the electron transport chain to make ATP, right? So that's why we eat. Because it's that potential chemical energy stored in the food. Our body has a way to break that down, put those electrons that are stored in those bonds of the food into these NADHs, FADH2s to drive the electron transport chain to get us ATP, right? Millions of molecules we're using every second. All right, so kind of this last question before we move on to fermentation. So why do we eat and breathe? So you should be able to answer that now on the molecular level, not just because it tastes good or we're gonna die if we don't. Those are both true, but at the cellular and molecular level, why do we have to take in food? Why do we have to breathe? So let's take a look at answering those questions. So all cellular processes, you have to have a lot of ATP, enough ATP to work. We don't store ATP. We don't have big like storehouses of ATP. So we have to constantly make it, right? Millions of molecules of ATP burned every second. So that's where these electrons come from. You have to be providing the food sources so we can strip them away and make those electron carriers to drive our electron transport chain. That's the food. That's why you have to eat. Why do we breathe? Oxygen was that final electron acceptor. So that helps to pull that oxygen molecule because it has a high electronegativity, helps to pull electrons through the electron transport chain. If you stop breathing, right, obviously you eventually die, but if you stop breathing, what happens is your cells use up all of their oxygen, gets converted into water, because that was that last step of the electron transport chain, and then there's nothing else to accept those electrons. So that carrier backs up, and then that carrier backs up, and then that carrier backs up, and then NADH cannot dump off its electrons because everything's halted. And if you cannot halt, if you halt your electron transport chain, you've stopped pumping your protons. right? If you stop pumping your protons, you're not maintaining that concentration gradient. It will eventually diffuse through the ATP synthase. You'll make your last little bit of ATP, and then you'll be at equilibrium. There's no potential energy if you don't have a gradient on that in the inner membrane space. So without oxygen, we back up our electron transport chain. We cannot maintain our concentration gradient of hydrogens, and our ATP synthase stops. And if we are not producing ATP, our cells die. So there's a little section in your book talking about cyanide poisoning and why it kills you in a few minutes is because cyanide goes and blocks that very last step of the electron transport chain, preventing oxygen from taking those electrons out of the chain. So cyanide is basically like holding your breath until you die, but a lot quicker because it affects all of your cells like instantly. So cyanide is a very toxic poison, and that's what it does. Is it stops your electron transport. All right, now kind of a transition into the last part of this chapter dealing with fermentation is our brain cells, for example, they can only do aerobic respiration. You have to have oxygen, you have to have glucose, they're super picky neurons. And so if you don't have the right amount of food or you don't have oxygen, your neurons are some of the first cells that go. Because they need that ATP to function at those high levels. Other cells in your body, like your skeletal muscles, for example, they can actually live for quite a while, function for several minutes, more than just the 3 minutes that neurons can function without oxygen before they start having permanent damage. But your muscles can actually run on what's called fermentation, kind of a lack of oxygen. Like think of the last time you were doing some really peak heavy exercises, you're breathing so hard, your muscles were burning. They were actually doing what we call lactic acid fermentation, where we are just not providing enough oxygen to fulfill all the requirements for aerobic respiration. So that's what we're going to move on to next to finish up the chapter, taking a look at kind of an incomplete breakdown of glucose by these processes of fermentation. Okay, so anaerobic means lack of oxygen. So oxygen poor environments, which could be in your muscles when you're exercising, or in a fermentation tank if you're making yogurt or beer or wine or something. Usually there's a lack of oxygen. So that pyruvate that's made at the end of glycolysis, if there's no oxygen to carry it into the mitochondria and finish the process like we just got done talking about, we can convert that pyruvate into a couple other products. Some organisms convert it into lactic acid or lactate. That's what our skeletal muscles can do. Or they can convert it into ethanol, which is a type of alcohol. It produces a little bit of ATP, just the two that come from the glycolysis, because it's a common pathway between cellular respiration and fermentation. But what it does is it allows a cell to recycle that NADH that's made in glycolysis back to NAD+ to keep the glycolysis process going. So let's take a look at the two types of fermentation. The first one is like what happens in our muscles. So this is lactic acid fermentation or lactate fermentation. So here we've taken our glucose, right? Here's our 6 carbons. We run it through glycolysis, right? So we're just having it be done in one arrow to our 2 pyruvates. Okay, so here's our 2 ATPs that we've made. Here's our 2 NADHs that we've made. So that's something that we just got done covering. In fermentation, we do not go to the acetyl-CoA step, Krebs electron transport chain. That is just for cellular respiration. But what we can do is we can actually physically change the pyruvates. We're not losing any carbon in a lactate fermentation, but we're getting 2 lactic acids. So they're saying 3 carbon molecules, but take a look at this reaction here. Okay, we generate the 2 NADHs in glycolysis. But if we're an organism and all we can do is glycolysis, we'd be quickly using up all of our NAD pluses and we wouldn't have anything to keep that glycolysis reaction going. So by kind of changing pyruvate into lactate, what we're doing is we're regenerating the NAD plus, which can come back over here and keep glycolysis going. So we're just getting the 2 net ATPs from glucose, but it's allowing us to keep glucose going by modifying that pyruvic acid into something else. So some products of lactic acid fermentation, so athletes, runners, if you do that peak level of activity, your muscles, your skeletal muscles will be producing lactic acid that then your body has to kind of filter out in your rest processes. Yogurt is made by lactic acid fermenting bacteria. Cheese is a product of lactic acid fermentation. Soy sauce, vinegar, there's lots of other industrial uses for bacteria, usually do lactic acid fermentation to make these useful products for us. It's just byproducts for these bacteria, but we can actually take advantage of that and have a nice little snack. The other type of fermentation is ethanol or alcohol fermentation. This is usually done by microbes, like yeast is the major microbe that can do ethanol fermentation. So again, we start off exactly the same way. So here's our 6-carbon glucose. We've made our ATPs, we've made our NADHs, and we've made our pyruvates, right? So they're like glycolysis. But in the lack of oxygen, when there's no oxygen, we can ferment it into slightly different products. So instead of lactic acid, we're producing ethanol, which is a 2-carbon alcohol, and we have carbon dioxide as a byproduct. And there's that NAD+. We're regenerating that to go back and keep that glycolysis going, because that's the only source of ATP for these organisms. And it's okay, because most of these lactic acid fermenters or the ethanol fermenters are single cell. They're not going to be burning millions and millions of ATPs per second. They just can survive on the 2 ATPs per glucose. Now, but you might be saying, well, Corey, you just said skeletal muscles burn millions of molecules of ATP per second, but some of them go to lactic acid fermentation when they're peak levels of activity. Well, that's true. How long can you maintain peak levels of activity? Not very long, right? You get fatigued, your muscles get fatigued. That's because you have run out of ATP. Your lactic acid is building up, the pH in your cells are going down, your enzymes aren't working very well, and so what you end up doing is getting muscle fatigue. So you can't maintain anaerobic fermentation of your muscles for an extended period of time like you can with an aerobic exercise. You can maintain that for a very long time. So let's take a look at some products of ethanol fermentation that humans have taken advantage of. So when you make bread, or if you make bread, the bread rises because of the carbon dioxide that's produced. And that helps to raise the bread because you use yeast, right? Yeast is the organism that does the fermentation of the sugars that you mix in, and then you get the dough to rise. Wine, beer, these are all examples of fermentation products where yeast turns the sugar that you put into your beginning mixture and converts it into alcohol. And, you know, big business, right? Especially here in Oregon. So that brings me to the last slide, is we actually have a state microbe. Oregon was the first state to vote and pass on a state microbe. Like we have state birds and state trees and songs and all this kind of stuff. We actually have our state microbe as yeast. It was passed in 2013 by our legislature, and so it is called Saccharomyces cerevisiae. It is the fermenter, this ethanol fermenter that gives us our brews, microbrews all over the state. So that is kind of a cool connection. All right, so that wraps up our chapter 8, which is glycolysis and cellular respiration. So hopefully by now you can kind of see those connections between photosynthesis and cellular respirations, reaching back and applying some of the information that we were learning back in chapters 5 and 6, and seeing how that content actually all plays together. All right, so that kind of also, I'll mention, wraps up kind of our chemistry, biochemistry kind of topics. So starting in chapter 9, our next chapter, we're going to be switching gears a little bit and heading into the nucleus to see what happens with all of our DNA as we take a look at cell division and heredity and gene expression making proteins. So I will see you next time. Bye.